I'm pleased that we have Stephen Benner with us. He's had been a distinguished professor of chemistry at the University of Florida. He's now with the Westheimer Institute of Science and Technology and the Foundation of Applied Molecular Evolution. I think it's almost fitting that we had Carl Zimmer give the introductory talk and Stephen give the closing talk. Carl's background in literature and journalism gives him the skills for looking in the past and looking at what has occurred. I think Stephen, with his background and PhD in chemistry from Harvard, gives him a perspective on thinking about the future. And I think his future almost borders sometime on science fiction. But I think truly innovative science should be almost like science fiction or magic. And so Stephen's has been sort of a polymath in the area of synthetic biology. He's, is one of the sort of helped launch the field. He synthesized the first enzyme. He's been very interested in sort of artificial expanded genetic systems, new artificial codons, new artificial nucleotides, and almost connected with that, he's very interested in astrobiology and looking for signatures of life out there. And so it's a pleasure to have him give our closing talk.
So thanks very much. Who actually saw Carl's presentation? Raise your hand. Who was worried by it? Oh, really? I mean, I was terribly worried by it because, as these first slides show, there is, of course, a counter anti-genome sentiment in biology. It's represented by Carl Woese's statement, the strange claim that some of the world's leading molecular biologists that the human genome is the holy grail of biology is a stunning example of a biology that has no genuine guiding vision. There is a secret, that is that the genome sequence of humans today tells us little more about the patient than the well-taken medical history does, especially if we add your parents' medical history to what we know. And there is this fact that, the errors in the data are often large compared with the signals in the genome that many would find interesting if you're interested in medical applications. But with Carl sitting up here and talking about yet another genome syndrome, if the journalists are onto us, we need to become actionably worried. And so I thought it would be important to wrap up this session with something of a informal response to what Carl said. Now, he's not here. He sent me a note saying he wouldn't show up, but I will send him this message at the end of it. Now, we need to become actionably worried. What I mean by that is not just to have another confab discussing errors and reasons why genome sequence interpretation is still quite far from us. But let's look at it from a perspective of culture. And as was mentioned, I'm a chemist, and you got to keep in mind that to us chemists, we're sort of troglodytes, and so a genome is really nothing more and nothing less than a statement about how carbon atoms, phosphorus atoms, nitrogen atoms, oxygen atoms, and hydrogen atoms are joined together in natural products that just happen to be involved in inheritance. And really the chemists will say, and nothing less than. This is actually not an insult, right? To a chemist, this is a good thing, right? This is what we do. And certainly in the German 19th century chemistry did this for a century, where they would take a plant, crush it, extract stuff, crystallize it to constant melting temperatures, melting points. Maybe you did this in your chemistry laboratory, and maybe you got a bad grade because your melting point was like 3 degrees too low. But there's a straight line between that and the human genome, and the German chemists in the 19th century, and the French, and to some extent the British chemists, very few American chemists, did this for a century without knowing that it would have any impact whatsoever about anything. And so there is sort of this view, if you live in the German-speaking world like I did for 10 years, that only the German culture could produce that kind of precision for that period of time without having any really obvious reward. But it's absolutely certain that the chemistry was done very well. And so chemists see no problem in assembling collections of natural product structures with no obvious immediate value. This is sort of our tradition. This is our culture. Of course, the chemistry culture is also relaxed about not having genomic natural product structures tell us anything about biology, because they never do, right? There is the chemist telling the biologist the structure, and the biologist's response is, so what? I mean, these are two structures of two molecules. If you are not trained as chemists, the most that the structure will, or not trained as biologists or endocrinologists perhaps, The most that these structures can tell you about biology is that they're not very soluble in water, right? And you would not know unless you had studied that this is a boy hormone and that's a girl hormone, right? So this is something that's not unusual to say, that natural product structures do not inform you about biology. And so it's a little surprising from a chemist perspective that people are disappointed that the genome sequences do not tell them about biology directly. They really shouldn't. They wouldn't expect them to. But one thing that was quite clear is that if the Germans published a natural product structure, it was right? This was their culture. And one of the problems that you have in the culture of genomics is that many features of that culture are antithetical to getting the structures right. And so we had, for example, for those of you who are old like me, you remember the Venter panic, right? That Craig was going to sequence and patent and own the human species. The public effort was terrified on that premise, and of course one of the things they required was that the release of sequences that you determine during the day by the close of business, right? That is not a way to build a culture of accuracy. That's actually a bad idea. I mean, admittedly, you don't want to be like crystallographers where you never release the coordinates, but you know, it's really not a good idea to do it quite so fast. We had the $10,000 and $1,000 genome disease. Now, of course, if you pay people by the word, you get books like War and Peace, okay? Now, I don't think it's wrong, okay? I mean, at some level, National Human Genome Research Institute's push in this direction, and Jeff Schloss should be given a lot of credit for actually doing a lot of effect. One of the very few cases of top-down at an NIH or public funding where you actually had really positive results of this. But today, even today, the major push of the NHGRI is reducing costs, not increasing quality, and not even talking about cost per unit quality or particular level of quality, and that's, of course, a problem. We have the X Prize syndrome. Okay, the contest has been going on now for 5 years, and I don't think they really have yet admitted they have no idea how to judge it. Right? This is you're gonna. I mean, my kid says you have to take Craig Venter's genome, change 1 out of every 10,000 bases, send it in, and let them figure out if they've got it right. Right? It's hard to actually score these things. Of course, the business plans are, you know, and you can sort of see it, is to sell instruments before the technology moves on, right? 454, Solid, Illumina, these organizations all had their day. Their goal was to just get as many instruments sold before somebody came up with a better idea, like PacBio or Ion Torrent or whoever, Oxford, I guess, nanopores is the next thing. No, all could have adopted chemistry that would have improved the quality of their product, but that was not the game. The game is it's just to get the thing out and get it sold, and because in about a year, somebody else is going to come along with a different machine which people are going to shift to. And of course, then finally, there's something which Carl, I'm afraid, as an outsider, he's able to see inside the deep bowels of genomics, the reward by press release. I couldn't see it. He's even said it, right? Right, you sequence a genome, you get your Nature Science paper, and you move on, okay? The reward to the scientist does not come by the use of that genome, and no penalty accrues from errors in that genome. And of course, the culture then, Carl again pointed this out, believed its own hype, revolutionized medicine forever. And for those of you who were here, you heard him ask, when does forever begin? And this, of course, is something that we can't let go without some degree of comment. So let me talk about comments. Let me talk about error, and let me talk about error vis-a-vis use. You know, because in 1980, when I was a graduating student, it was clear to all of us that total gene sequencing was going to occur. It's only a matter of time. George Church tells me that he took until 1985 to realize that. I don't believe that. I think it was sort of obvious earlier, and that's just because it's the natural trend of things, right? So, I don't. I'm afraid that might give you the impression that I spent 30 years of my career waiting for the JGI to start doing genome sequencing. I don't want to give you the impression that we have been not doing anything in the meantime, but yeah, so one of the things we do want to be prepared for it, so we certainly did a lot of total gene synthesis because DNA synthesis is what chemists do, and it's quite clear that synthetic DNA is going to be very important for whole genome sequencing. One of the things our really goal is what we were calling it's planetary biology. If you have the genome of every organism on the planet, or, you know, failing that, one representative of every species on the planet, or failing that, just some dense distribution across the tree, you can start doing across-planet correlations. You can start talking about grasses as they interact with animals that eat the grasses as opposed to the bacteria now in the stomach of the animals that eat the grasses. as opposed to the humans who eat the animals, who have the bacteria in the stomach that eats the grasses. And that's obviously the future when you have actually a lot of genome sequences. So it's not the yet another genome syndrome, that's actually the plan, and we'll talk to Carl about this in a minute. But the second thing we really were very interested in doing, and I'll talk a lot about this today, is to try to bring experimental method to this analysis of cross-planet genome sequences, because remember, a lot of those explanations, models, and analyses are historical in nature. They're evolutionary in nature. We're saying that genomes are the way they are, ecosystems are the way they are, organismic interactions are the way they are because they evolved to be so through events in the historical past. Now, many people, especially physical scientists, many of you in the audience, don't think that natural historians or hypotheses that come from natural history are testable because they occurred. They talk about things that occurred in the past, and you can't build a time machine. And so many physical scientists, and I must say, myself was brought up this way, was to think that natural historians aren't really scientists at all, right? They're just sort of storytellers. And I'll get to that in just a minute. But, you know, I mean, one of the things we're also going to reflect to is this master catalog we actually did develop, and actually was a product. It sold about $3.5 million worth of software in the late 1990s, early 2000s, where we're going to construct databases which organize evolutionarily the genome. Not the genome, but all genomes in a database. And you can go back and read some of this stuff about planetary biology. There was a paper in 2002 in Science on the subject. So our goal now is to talk about error and what the error rates should be sought in whole genome sequences to support this idea of cross-planet organism interactions. And I'll talk to you a little bit about that. Okay, planetary biology. I'm sure all of you are tired of hyphenated biologies, right? Systems biology, synthetic biology, astrobiology, planetary biology. But here you are looking on the left of this slide at a temperature course of history on Earth. The vertical scale here is about 70 degrees centigrade, but what you're actually looking at is oxygen 18, oxygen 16 fractionations in sediments. So this is actually a proxy for temperature. If I had extended this back, well, this is the age of the dinosaurs. This is the Cretaceous. If I had extended this back earlier in the age of the dinosaurs into the Jurassic, the climate was actually relatively high, hot, and stable. What has happened, especially since the end of the Eocene through the Oligocene, the Miocene, and then, of course, in the Pleistocene, is that the planet's climate has gone to hell in a handbasket, okay? For those of you who are worried about global warming, I mean, you'll understand the paleontologist sides of us. Don't worry about this at all. The planet's problem has been cooling, and this has driven you out of things, well, it's actually driven many things, but it's driven physiology in microbes and plants and animals, as well as in their genomics. Here you are with the jungle, verdant Garden of Eden of the Eocene gone, being driven out into the savanna. Just for the record, there was no sea level ice anywhere on Earth in the Eocene 40 million years ago, not even Antarctica, and you know that because if you go to Antarctica and dig around in the sediments off the continent's coast, you will find leaves. You'll find deciduous tree leaves there. And so this is, by the way, this is for those of you who believe climate change models, and I know there's some people in the room who do. None of them account for any of this at all. We don't really understand this. There are hypotheses. There's hypotheses based on continental drift that maybe Australia in the Miocene drifted away from Antarctica, setting up a circumpolar current which maybe isolated Antarctica from the currents that bring heat from the equator to the poles. That's a view. Some people talk about the ice ages as a consequence of the Panama Plate sailing in between North and South America, therefore creating a pole-to-pole or nearly pole-to-pole landmass that separates the Atlantic and the Pacific. There are people who talk about this. But at the end of the day, almost everything that people worry about today in terms of climate change is small to the point of being negligible. I guess I can't go backwards in this talk, but if I could, I would. But the bottom line is it's small compared to being negligible, what I showed you in that previous drop, and that's a big problem. Well, there's this problem. These are what we call just so stories. There are problems with these just so stories. Remember, science is an intellectual process that embodies a mechanism to prevent us scientists from always coming to the conclusion that we set out to come to, right? Most of the time that mechanism fails. But, you know, the goal here is to try to get something to confirm or deny these just so stories, because a just so story is one of these stories that would be just as good if the facts were the opposite. We could come up with something that would explain them. And so it would be very nice to bring experimental methods to bear on any of the historical models I've just rattled off to you, including the climate ones. And for that, we'll talk a little bit about paleogenetics in a minute. So let me start now at the beginning of that drop-off of temperature. Okay, we're in the Cretaceous. You're looking at the rise of angiosperms. This is something gotten by my colleague David Dilcher at the University of Florida. It's actually a plant coming out at the border between the Jurassic and the Cretaceous. This is probably not an angiosperm, by the way, a flowering, fruiting plant. It is actually assigned that in this particular article. It's certainly a very, very primitive angiosperm if it is one. But what's clear is as the Cretaceous moved on, these angiosperms did develop. There are some fruits that you can actually get from the Cretaceous. This is actually a magnolia fruit from the British Columbia area. This, of course, is the modern grape. But I put this up here to remind you that, yeah, once you have the plants evolving, the entire ecosystem evolves, because angiosperms are going to take over the planet and have quite a different view of animals that associate with them than conifers do. So conifers basically don't want you to eat them. What angiosperms do is offer you a treat to eat them with a seed so that you disperse it. But of course, across the genome in a planetary biology sense is not just the plants and the animals that eat the plants, the birds, that is dinosaurs, who went to live in the trees, which unlike conifers have spreading branches to welcome you as a bird in, or as you as a primate with opposable thumb in. But also, of course, the yeast are responding to this. And this is a fungal story because the yeast are going to invent alcohol fermentation when these fermentable fleshy fruits become available. Because by making alcohol, the yeast is going to defend this rich carbohydrate source for itself, because alcohol, as you all know, is toxic and it kills off competing microorganisms. And so when you have a fully fermented grape in the form of a wine, that grape is sterile with respect to everything else but the yeast, to a first approximation. So this should leave a record in the yeast genome. It should also have, by the way, and it does leave a record in the fruit fly genome and in the plant genome as well. But let me just talk about the yeast briefly, because we'll now go looking in the whole genome sequence. And remember, that's the game here. If I had to go look for individual items in the gene sequence, sequencing one at a time, I would never find this. But when we get the whole yeast genome, we can start asking questions, and Mike Lynch did this in 2000, looking for whether or not you could find events in the whole genome that reflected yeast adopting this new lifestyle about 80 million years ago to make fermentation alcohol once fermentable fruits became available to it in its ecosystem. Now, Mike actually used the wrong metric. He actually is using a metric which is looking at substitutions per silent sites. And actually, when you use the wrong metric, you miss the interesting result, because silent sites are these kinds of things. You have A, T, G, and C, but of course you have both transversions. That's where a purine on the left becomes a pyrimidine on the right, and of course each of those arrows, 1, 2, 3, 4, 5, 6 on the diagonal, 7, 8 at the bottom, there are 8 transversion rate processes. And in principle, if you're a chemist, there are 8 transversion rate constants. And of course there are transitions, which are the purines going to another purine. There are 2 rate constants there, or pyrimidine going to another pyrimidine, T to C. There are 2 rate constants there. And there's no reason to believe that the rate constants of any of those are the same. In fact, they're not. They're actually somewhat different. And so if you start lumping them all together, you tend to smear out the events. And here these events are gene duplications in the yeast genome as clocked using substitutions per silent site. But you can separate these out. I mean, we have separated the green sites out from the red sites. The green arrows, of course, are transitions. There actually are 4 rate constants. They tend to be rather similar and quite a bit faster than the transversions, which are the rate constants indicated by the red arrows. What's very nice about 2-fold redundant codon sites, like those encoding lysine or glutamine or glutamic acid or histidine or cysteine, for those of you who know the code, is that if you have a cysteine that's conserved, what will happen is that the 3rd position site will have an approach to equilibrium process whereby Everything starts off as a T in the cysteine codon in particular, and will gradually become C until after you had equilibration, there'll be equal amounts of C or T, or the ratio of the two will be determined by codon bias. And you can actually write out relatively simple exponential approaches to equilibrium equations, and you can actually get what we call the transition redundant exchange clock, which we call that simply because it gave the acronym T-Rex. All right, people, this audience does not appreciate acronyms. All right. So what you in fact do is align the genes, you identify sites where the amino acids that are encoded by twofold redundant codon systems are conserved. You examine the silent sites in those positions, you calculate the fraction of the identical sites, that's an F2 value, and you determine the time of divergence from rate constant. You do the exact same thing, I won't go through that because everybody knows that. You do the exact same thing. What you discover is that there's a lot of gene duplication apparent, and that could also be gene conversion, which I will not talk about. Recently, there's a lot that goes on a very long time ago, and that's this cluster to the left. Those is where the threefold or twofold redundant codon system has equilibrated. You cannot measure a time at all. But there's an episode of about 15 gene duplications that occurs about 80 million years ago, which is the time when fermentation emerged. Well, it's not just that. That would be sort of an observation. It turns out that the genes that are undergoing duplication in that episode are not randomly chosen from the metabolome. It turns out that there are glucose import enzymes. There are enzymes involved in rate determining steps of the alcohol fermentation pathway. There is an enzyme duplication that allows thymine to be transported more efficiently, as well as a duplication that involves the pyruvate decarboxylase that uses thymine and is non-oxidative. And there's also a duplication of these two alcohol dehydrogenase genes, ADH1 and ADH2. And for those of you who know about yeast, this is ADH1 is constitutive. It's turned on in high quantities, got strong codon usage bias, and it's making all the alcohol. And then when all the alcohol, all the sugar is converted to alcohol, then yeast turns on ADH2 and sits there and drinks the beer if it has oxygen available to it. Well, okay. So certainly this history of duplication, and which you can come upon only by a whole genome sequence, is consistent with a new lifestyle for yeast that is contemporaneous with the formation of the fermentable fruits, the emergence of fermentable fruits on the planet. But of course, now you can do an experiment because you have the ability to infer the sequences of the ancient alcohol dehydrogenases, use recombinant DNA technology to bring them back to life, and see what kinds of kinetic behaviors they have and what kinds of roles they play. Now, for those of you who are into what we call in the business perfect enzyme theory, you know that there is a rule. I mean, enzymes all catalyze reactions in both directions. They don't perturb the equilibrium, but when you're out of equilibrium, enzymes will evolve to be optimal for the direction in which they catalyze a reaction. And you can judge this, for example, by looking at the Km for one of the substrates. I've just put here the Km for ethanol. For ADH1, which makes ethanol, the Km is very, very high. That prevents it from not being product inhibited. But for ADH2, which is turned on when ADH1 has completed the conversion of all glucose to alcohol, and the yeast now wants to sit back and drink beer, that has a very low Km for ethanol. And so, of course, what you can now do is collect sequence data, and of course it would have been very nice if there were whole genome sequences for all of the Clavaromyces and Saccharomyces and Pischia and Bayanus and all the other species that are on this tree. They were not. Most of these were collected by individual sequencing. And you can infer the sequences of ancient proteins by looking at the sequences of the derived proteins. I've actually put up one site here which actually is a source of ambiguity, and you can see why it's a source of ambiguity, because you're actually seeing homoplasy in this site, that is parallel replicated amino acid replacement. This is a site that's not freely evolving to accept all 20 amino acids, but there's some evolutionary constraint on what amino acid is present at this site. But our case is when we have ambiguity here, we're going to resurrect the last common ancestor of alcohol dehydrogenase 1 and alcohol dehydrogenase 2, represented roughly by that star on the plot right below the red line. And we're going to, when we have ambiguity at a site, what we're going to do is make all possible combinations and permutations. In fact, in this resurrection here, there are two sites that are twofold ambiguous, And there's one site that's threefold ambiguous, so we must make 2 times 2 times 3, or 12 different ancestral gene candidates. And we hope that now when we measure the KM on these ancient enzymes after we bring them back to life using recombinant DNA technology, we certainly hope that they will give a more consistent story. In fact, some of these enzymes don't get expressed. They're pathological, and I'm assuming that those combinations are not the ones that were actually present in the ancient yeast. But for the most part, the ones that are active, you actually have KMs that are in the 7,000 micromolar or 8,000 micromolar to 20,000 micromolar. These are enzymes that are actually making ethanol. They're tenfold. In all cases, the KM is higher by a factor of 10 or so than the KM of an enzyme that consumes alcohol. So the inference is that the ancestral yeast and the ancestral alcohol dehydrogenase made but did not eat alcohol, ethanol. So that's kind of cool. That means the ancestor of ADH1 and ADH2 acts like ADH1, which means that you don't have this reverse reaction, which means that the ancient yeast is never getting itself beer to drink. And in fact, for those of you who remember the metabolic pathways you were forced to memorize, you'll remember that in glycolysis, at this step, glyceraldehyde phosphate dehydrogenase, you make an NADH, which you have to recycle. You will remember from your own courses that you'd actually recycle that using lactate dehydrogenase. This is why you make lactate in your muscles. Here, of course, the argument is that the ancient yeast, before it's starting to use this fermentation strategy where you accumulate alcohol, kill off competing species, and later drink the alcohol, here the yeast is just using this last step to recycle the cofactor. So that's great. So you're looking at what we mean by planetary biology. It's a signature in the yeast genome of events actually in fruits that is correlated in time by some combination of clocks and fossil records. And that's work that actually Mike Thompson and Eric Gaucher. Mike is now at University of Georgia, and Eric is actually Georgia Tech. They just moved north. Now that's here. That's where fermentation is starting. That's up where you have a Garden of Eden, as Dan Prothero talks about it, warm temperatures everywhere. And now, of course, for some reason, you start to have planetary cooling. And of course, what happens is that grasslands open in the Oligocene, forests went away, the Garden of Eden was lost, and grass is a lousy food. Who has tried to eat grass? Raise your hand. This is, okay, well, you're looking thinner for the effort. So, of course, across the biosphere, there's repercussions among grass eaters. And of course, the grass eaters that emerged are the ruminants. Because at the Eocene, there was maybe one species of ruminant that we know of in the fossil record, Diacodexis. Actually, he was not even a ruminant. He was an artiodactyl. He had a cloven hoof. There were maybe 250 species of odd-toed ungulates, the animals that are now represented by just 3 or 4 species, the horses and the rhinoceroses and the tapirs. So you're going to see a massive extinction of the parasodactyls, as we call them, the rhinoceroses and the like, the brontotheres. 250 species go down to 3. 99% of them are gone, roughly. And replaced in the large animal grass-eating ecological niche of the ruminants, they collect grass, they feed it to their first stomach. As you may know, bacteria in the first stomach of a ruminant then converts the grass into low molecular weight fatty acids. This is why a goat smells like a goat, or the capric and caproic acids. The bacteria are then passed to the second stomach where they are digested. So the ox eats bacteria. He does not eat grass. He collects the bacteria. And of course, to eat bacteria requires a special set of ruminant enzymes, the lysozyme to break down their cell walls. The protease is special for bacterial proteins. Ribonucleases, because bacteria have abundant RNA. About 20% of the nitrogen intake of a typical ruminant is in the form of RNA. And you, in contrast, have essentially no RNA in the diet. I mean, your nitrogen doesn't come from RNA to a first approximation. It comes from protein. But a genomic signature of climate change should be in the ruminants. So you can go looking for it. In fact, you can actually see the enzyme that degrades the RNA coming in from bacteria is present in ruminants like ox and buffalo and sheep and camel and deer. Not so much in pig, a related artiodactyl, but not a ruminant artiodactyl. Of course, is really absent in you. And so the emergence of ruminant ribonuclease should correlate with the climate change, just if Barnard's just so story is true. Well, you, of course, can go back and now do the same game. And by the way, all of these sequences, none of them comes from the Eland Genome Sequencing Project or the River Buffalo Sequencing Project, but we would have liked to have had it come so, right? Because, of course, what we have done here is sequence these, genes all ourselves, which means we had to get a piece of eland. And eland steak is not something that is sold at Publix. But you can infer the sequences of ancient proteins, ancient ribonucleases. And what's kind of amusing about this is that you can now make a measurement. And all these paleogenetic studies have a measurement, right? You have to decide a feature of the protein which will tell you whether it's digestive or not. Here we're looking about the feature of whether the protein is itself easily digested, because if you put an enzyme into your stomach to digest food, you don't want it to be digested along with the food. And so in fact this ribonuclease that's 4 million years old is just as stable against digestion as the modern ribonucleases. And that's true if you go back all the way to the divergence of giraffe and pronghorn antelope, but if you go back even more ancient to the ancestral enzymes from, say, the species like Diacodexis or older, these are not ruminants. They're the ancestors of the non-ruminant artiodactyls, the hippo and the pig, and to some extent the whales, by the way. What you discover is all of a sudden the ancient proteins no longer behave like digestive enzymes. In particular, they're not themselves stable against digestion. They actually have new activity to bind to double-stranded nucleic acid, which is not displayed by any of the derived enzymes in the digestive tract. And all of a sudden, you see that the origin of ruminants, right, correlates with the behavior of the enzyme involved in ruminant digestion, as Barnard's hypothesis proposed. It's supported by experiment, and it correlates across the planet with the grasses emerging and the climate changing. So what about the impact on primates, right? Because that's, of course, what we're all interested in. When in primate evolution did this climate change really begin to have an impact? Now we know this somewhat from the record, right? Because the first thing that happened when primates lost their Garden of Eden, their forest, is that their range shrank. There were primates, thanks. There were primates all over the place in Europe and beyond. I don't know if there are any primates in Antarctica, but there were certainly primates way outside the narrow range in Central Africa and Mesoamerica, Latin America, Central America, and the Amazonia area. Because what happened basically with the range shrank and many Miocene apes just became extinct. Part of the reason was because you made a mistake in your genome a couple million years earlier of getting rid of the gene for making vitamin C. That turned out to be a huge blunder once the planet decided it wanted to try to kill you. What you have done, and you actually have done so in the baboon lineage as well, is created the gene for, you've lost the gene for degrading urate, and the argument is that urate stands in as a bulk antioxidant where an ascorbate fails. Of course, urate also is what gives you gout, so there's an associated disadvantage of having a slightly less soluble antioxidant. But never mind, the bottom line is that we can connect enzyme behavior, and we can talk about alcohol in particular, because there you are in the Paleocene. By the way, that fossil is known by a couple of teeth, a jawbone and a femur, so that sour expression is inferred by artists. Of course, you are now coming out to Ardipithecus ramidus, this is Ardi, as people call it. We would like to know when in the sequence. We know yeast is making alcohol. We actually know fruit flies are going and looking at alcohol roughly at the same time it is first made, but it's not quite clear when in the progression of primates you started to interact with this new substance on Earth arising from a new lifestyle for plants. So actually, there's kind of an amusing story here. This is actually a two-step pathway. For those of you who think that alcohol is toxic, you're right, but of course the oxidation product of alcohol, that is acetaldehyde, is really toxic, right? So it's not just that you need an enzyme to oxidize the alcohol to the aldehyde, but you also need the enzyme to oxidize the aldehyde to the carboxylic acid. You can't just do something trivial if you want to try to drink the beer, right? That is, overexpress the enzymes that you have, because it turns out there are lots of alcohols, not the least of which is retinol and retinal, which of course you use in your eyes, and of course retinoic acid, which is a transcription regulatory factor. And there's a fetal alcohol syndrome story built into all this. So if you just all of a sudden start drinking alcohol and just to overexpress the enzyme that you oxidizes retinol, retinal, or to retinoic acid, what you end up doing is screwing up all the other metabolism that you have going on here. You actually have to change the structure of the protein to become a little bit more selective. By the way, forget yeast and its duplications. You have actually ADH1, ADH2, ADH3, ADH4, ADH5, ADH6, and ADH7, only a few of which are, well, some of which are more active than others. ADH1 actually has ADH1A, B, and C. That's a gene duplication that's occurring right before New World and Old World primates split, by the way. Ardipithecus already has 3 ADH1s, an ADH1, ADH2, and ADH3. ADH4 actually is single orthology cross. I'll talk about that in a minute. But so the sequence of RD's 3 forms of alcohol dehydrogenases are known to us by inference, by resurrection, or by Bayesian analysis of the derived sequences. But of course, that doesn't mean ethanol, right? Because I've already mentioned there are lots of other alcohols. I just put up here the structures of all the alcohols that people have invoked one time or another as substrates for alcohol dehydrogenases in various tissues. I mean, there are alcohols that are coming from the oxidative degradation of lipids. There's alcohols that are coming from histidinol. There's alcohols, of course, that are coming from plants. And remember, one of the other things that angiosperms do a lot is make a lot of these funny terpene products, and of course, retinol, here's a structure, one of them, but also cinnamyl alcohol and half a dozen other terpenes and aromatic species. So the fact that you have a lot of gene duplication going on in the history of primate lineages doesn't mean that you're drinking alcohol all of a sudden, or even at all. It could very well be in a response to any of a number of other things in your diet. So it becomes actually quite important to do some other information to try to add to this story. Now, one thing you can do is do structural biology. Here is actually the crystal structure of one of the 2 subunits. This is a dimeric enzyme. The yellow is NADH, the green is ethanol, and the red is a contact site 42, which is where ethanol binds to the enzyme, right? So what you can now do is go back and analyze the particular structure. But remember, there's more to a site than either conserved or variable, right? Here I've given you different patterns of variation within a hypothetical site to illustrate this point. You can read about it in all these references that we've published, right? I mean, this site can have valine or alanines is the same number of valines and alanine in the left tree, and the same number of greens and reds in the left tree as in the right tree. But as you can see here, it's an indication of conservation with a single change, the ancestor here being red, the ancestor here being red. Whereas here you're having homoplasia. You're having, many times valine red goes to alanine green all across that tree. So they're making 2 very different statements about conservation and purifying selection as it operates on the amino acid at that site. In the bottom cases, you've got 2 trees which see a lot of variation, but you'll notice in the left tree the variation is distributed across the family, but in the right tree, you have the variation in the right subbranches, not in the left subbranches. This is called heterotachy, and what it indicates actually is that somewhere up here at the top, there's been a change in function because a functional constraint that is conserving a valine on the left-hand branch is no longer operating to conserve the amino acid on the right. So this is a statement of change in function along that branch in the tree. Now, if you go back to alcohol dehydrogenase and just add crystallographic studies to that thing, right? So you look for the sites that where they're conserved in the fungal enzymes but variable in the mammalian enzymes, conserved in the fungal enzymes and variable in the mammalian enzymes, and you put those sites at a crystal structure, you discover that all of them cluster in the substrate binding site, which is just another way of you're saying that yes, in the mammalian enzymes, but not in the yeast enzymes, which are acting on alcohol, and only alcohol. In the mammalian enzymes and the liver enzymes, in any case, you're changing substrate specificity. This is a sign of adaptive change. So that's cool, but fortunately it turns out, and it's complicated, and it would take me another hour to describe ADH1 and its variants, but ADH4 is the enzyme in your stomach and esophagus. There's only one ortholog across this tree, and actually what we're doing now is trying to build a we would have a tree which will show the orthology and to understand the evolution of ADH4, because that's actually the first enzyme to see the beer. Now, it would have been nice, by the way, I've got on branches for those of you in the front, you can see it. I have on each of these individual branches down here what amino acids are changing along those branches. It would have been nice to have gotten these from whole genomes. So to tell Carl that yet another genome would have been very nice for, the tamarin or the tree shrew. Actually, there is a genome for the tree shrew, but it's not a very good one, as it turns out. But we had to go do each of these sequences for the most part ourselves, which means we had to get the tissue. And I assure you, you have to make friends with many, many zoos to get monkey tissues from this. So that no one is going to kill a monkey for you, right? This is clear. So what you'll notice, and I just put some raw data up here collected by Matt Carrigan and Carol Fry and Blair Ekman. One of these enzymes, this is just a raw trace. One of these enzymes is active. A lot of enzymes are moderate active with ethanol. And it turns out that where the enzymes become active, from ethanol goes from low activity to high activity down this branch of the tree. And for those of you in the back who can't see that, this is the branch of the tree by which the last common ancestor of the orangutan and you evolves to become the last common ancestor of you, the gorilla, and the chimp. So all of a sudden, what you're saying here is that although Ardipithecus and all primates, actually for that matter, are living in a world of ethanol back to about here, that is, back to about here is where the world of ethanol, you're not partaking of ethanol until very, very recently. And that's actually an interesting statement. It's actually kind of amusing that the tree shrew is an exception to that. It actually has it actually drinks fermenting fruits and has a high alcohol activity as well. So that's actually an example of homoplasy in terms of function. So this leads to the question, did Artie party hardy? And the answer is it seems so, but perhaps an anticipated change. Right, well, we have what we infer, and of course what we infer next is that males are beginning to enter into relationships with fraternity. But this is what we can do with planetary biology when it is supported by complete whole genome sequences. This, of course, is the answer to Carl at one level, also understood by natural product chemists who really don't care if we collect a lot of stuff and don't learn anything more than just that the person's eyes were brown. There's a lot of examples now in paleogenetics. One of my favorites is actually not one of ours. This was actually done by Mikhail Matz, who actually resurrected ancient fluorescent proteins and painted a little evolutionary tree showing the evolution of the fluorescent color on a petri dish. This has been going on for some time now. And now, I kept saying to you we did not use complete genome sequences, and you might want to ask me why, and this gets me back to the point of error. There is actually a tree genome sequence. We're not able to use it. Why? Because it had 6 of the 7 exons. It didn't have the 7th. It was missing one. We have actually had for many, many years something called the master catalog, which has pre-computed evolutionary models for all the families of all proteins on Earth. It has a multiple sequence alignment of tree, reconstructed ancestral sequences, all the rest of it. We've sold it. There's actually a paper in 2000 where you can read about its details. But we've come back and asked whether we could build the master. In the old days, we just built it from a reliable database, SwissPro, a very reliable database. It's German, actually, but never mind. Could we build it from today's databases? Well, the answer is we went back and tried, most recently from 18 chordate genomes deemed to be the most usefully reliable. And of course, what you get is a family like this, where all of a sudden most of them are great fully sequenced, but there's the macaque sequence. It's a fragment. So, you go on, you say, okay, there's the macaque, there's the fragment, by the way, in the multiple sequence alignment. And of course, it screws up the tree, right? Right, usual. Still, other families are entirely missing orthologs. Now, you never know whether that could be right? The gene could have been lost. But we've gone back and looked up, all sorts of things, right? The perfect family, and this is the master catalog built from these 18 genome. So the perfect family has 18 orthologs, right? Of course, duplications, some sublineage will add to this. Real gene loss will subtract from it, but so does error. And so you can actually see there are a lot of families with 18, a lot of families with 36, and so on. You can actually see the number of species, the number of taxa that go in here actually does give you input there. But we have 569 families with exactly 18 members. Okay, of these, about 10% are truncates. Of these, about half of them, the missing sequences, could not be found by returning to the primary data. So that's the primary data are missing it in half of this set of, quote, perfect families. Now, some of the DNA could be found if you went back to the primary data and could be recovered. And of course, that's the first case is a failure to sequence the DNA, and the second case is a failure to find the DNA. Of course, we can go through this for hours. Only 130 of the 569 families were perfect. You had a lot of 18 minus 1 families and 18 minus 2 families. And I can go back and tell you what fraction of those, they just missed the sequence in the database, or whether we can go back to the primary data and find the sequence, which meant it was missed in the assembly or in the gene finding. This is fine. This is all, by the way, put together by Kevin Bradley, except the whole genome sequencing is being sold because it can tell us what's not present. And of course, if what's not present is because it's been missed at one of various levels in the process of the database, that's a problem. Well, there's hope. There's hope for new reagents. I will not even give this talk to you. You can go to a catalog and see how we can do all sorts of things that are available. Reagents are available to improve the output of genome sequence analysis. You just can't find them on the Illumina instruments. There are tools to exploit quality genome sequence data with medical impact. That's an answer to Carl's question. They're just not on the NCBI webpage. But you can go, we sell some of these. You can actually go back to www.ffame.org and actually dig them up if you want to see whether you can try to improve some of your analyses using these. But Carl is not quite right. There's a lot of secrets in this business. There's a lot of problems, and there's right now, there are actually very few genomes that are actually completed to a level adequate to support planetary biology. We're worried that the science journalists are onto us, right? We need to become more actionably worried. There are reagents to improve outcomes, especially sample bias in what you sequence. There are tools to clean up the database, and planetary analysis will indeed, I think, revolutionize biology. But to do so, we really must change the culture a little bit to realize this vision by removing a little bit the error-tolerant funding reward structures and business plans that are dominant in the culture of the field. So with that, let me stop. Thank you for your attention. I'll be happy to answer any questions you might have.